faithfully delivering value by interpreting the world's great books. Hello, this is Sally's Book Talk. Today we're exploring the book Virus Planet. Our Earth is truly a virus planet. From land to sea, viruses are everywhere around us, constantly impacting our health. For instance, just in our lungs, there are an average of 174 types of viruses, and we're unfamiliar with 90% of them. Besides causing terrible diseases, viruses can also help cure illnesses. Even the oxygen in our respiratory tract is closely tied to viruses. Today's Virus Planet will share the inseparable stories between viruses and humans. The author Carl Zimmer is a renowned American science writer who teaches science and environmental writing at Yale. He joined the Virus Planet Project by the National Institutes of Health, aimed at popularizing virus knowledge, e.g. 1 KTFA. This book is one outcome of that project. It explains how viruses spread and cause diseases, while also showing that humans can't live without viruses. The Washington Post reviewed the book, saying, "In this 100-plus page work, Carl Zimmer unveils the veil of daily life and reshapes our fundamental understanding of viruses." Carl Zimmer will all be active. Next, I'll guide you through 3 parts to understand viruses. First, what is a virus? How does it make people sick? Second, how do viruses spread between different species? Third, why can't we live without viruses? First, in part 1, we'll explore what a virus is. How does it make people sick? Although viruses have always shadowed humans, virology is a young science. Viruses weren't discovered until 1892. Back then, Dutch tobacco farms were devastated by a disease, wiping out harvests. People suspected a bacterium was to blame. But bacteria from the diseased tobacco juice couldn't infect healthy plants. Folks wondered, could something smaller than bacteria be causing it? So they filtered out cells and bacteria from the juice and injected the remaining liquid into healthy tobacco. Then the tobacco got sick. This pathogen was the first virus discovered by humans, the tobacco mosaic virus. It's not only tiny, but also incredibly tough. Even if you boil virus-laden liquid, the virus remains unharmed. Soak filter paper in the virus solution, let it dry completely, then 3 months later, soak it in water. That water can still infect tobacco plants. To biologists back then, viruses represented a completely new life form. Tiny, structurally simple, non-cellular organisms that must parasitize cells. Viruses are entirely different from bacteria, another microbe. First, viruses are much smaller. Regular microscopes can see bacteria but not viruses. Only higher magnification electron microscopes can reveal viruses. Second, viruses have a simple structure. Bacteria are single-cell organisms, while viruses don't even possess cells. Essentially, it's just a protein shell containing a handful of genes. Because viruses are so structurally basic, scientists still debate whether they can even be considered life. Third, viruses parasitize cells. Since bacteria are cellular organisms, viruses can also infect them. Viruses inject genes and proteins into host cells, turning them into replication factories. A single virus can replicate thousands of copies within one cell in just a day. Our most direct understanding of viruses is that they make people sick. Different viruses operate in vastly different ways. Here, I'll introduce 2 representative viruses. The first is rhinovirus. Rhino meaning nose, which triggers common colds and asthma. Though colds are minor illnesses, they're exhausting. It's estimated that each person spends a full year battling colds in their lifetime. Plus, with over 120 identified rhinovirus strains, medications or vaccines only target specific types. Once a virus mutates, even if it just changes its protein code, medications or vaccines can no longer recognize it. Rhinovirus invades cells in the nose, throat, or lungs, replicates inside them, then newly formed viruses burst out of the cells. They then infect more cells. When rhinovirus attacks these areas, nearby immune cells rush to the site, destroying the virus, but causing inflammation. Infected areas secrete mucus and show swelling, redness, and itching.
a protective immune response.
Without an inflammatory response, we'd suffer continuous infections. So, to recover from a cold, we need both the immune system to eliminate the virus and then calm itself down. That's why colds seem so hard to cure. But from another angle, rhinoviruses train our immune systems to avoid overreacting to minor irritants in the future. Research shows childhood infections with mild viruses like rhinovirus reduce adulthood risks of allergies and chronic intestinal diseases. Moving beyond rhinovirus, HPV is a virus that can cause cancer. HPV, or human papillomavirus, causes skin hyperplasia symptoms like wart-like growths. An Indonesian boy with HPV had nearly 6 kilograms of hyperplastic tissue surgically removed. Moreover, HPV causes cervical cancer, the third leading cancer killer of women after breast and lung cancer. HPV specializes in infecting epithelial cells that line skin and body cavities. As our skin renews itself, when outermost cells die, underlying cells replace them. When HPV infects epithelial cells, the virus travels outward, with these migrating cells toward the skin surface. Cells near the surface rupture, releasing viruses that infect new hosts through skin or sexual contact. Occasionally, during replication, viral DNA integrates into host DNA. Instead of dying, these cells form tumors, causing cervical cancer. A 2014 international study found 71 out of 103 healthy individuals carried HPV, a 69% infection rate. But don't panic. Carrying HPV doesn't guarantee cancer. Of over 100 known HPV types, only 14 are high risk. Most others are low risk. While 30 million U.S. women carry HPV, only about 13,000 develop cervical cancer annually. More importantly, cervical cancer is currently the only cancer preventable by vaccine. In 2006, the first HPV vaccine launched in the U.S. and Europe. It contains HPV's shell proteins, training the immune system to recognize the virus. Once infected, the immune system can immediately counterattack and eliminate the virus. That concludes part 1. We've covered basic virus facts and how rhinovirus and HPV cause illnesses. Next, in part 2, we'll explore how viruses cross species barriers. Studies show over 70% of emerging infectious diseases originate from animals, like AIDS, Ebola, and SARS, viruses that entered humanity just in the last century. How do these newly emerged viruses jump to humans? Take influenza. Unlike common colds, it's vastly more contagious and deadly. While cold-causing rhinovirus is human-specific, flu viruses originate in birds. All human-infecting flu strains trace back to birds. For flu viruses to jump from birds to humans, first, they must enter the human body. Originally, flu viruses had nothing to do with respiratory systems. In birds, flu viruses reside not in respiratory tracts, but in digestive systems. Viruses hide in bird droppings, so poultry farm or slaughterhouse workers risk infection through contact with chicken feces. To enter cells, viruses need receptors, binding substances on cell membranes. Coincidentally, receptors on human respiratory cells closely resemble those on bird digestive cells. Thus, viruses feel at home away from home, logically entering human airways. But entry isn't enough. Viruses must adapt to human physiology. Humans differ vastly from birds, like having lower body temperatures, making viruses uncomfortable. Even after infecting humans, person-to-person spread is rare, but some viruses mutate to adapt and achieve transmission. When flu viruses infect new hosts, sometimes other viruses already inhabit those cells. Then these distinct viruses must coexist and replicate within the same cell. The next viral generation may then carry genetic material from both parent viruses. This genetic mixing phenomenon is called reassortment. Through reassortment, flu viruses create new genetic combinations, gaining new characteristics. New viral strains emerge.
Having never circulated in humans before, no one has antibodies, making it unstoppable with nothing to slow its spread.
Take the H1N1 flu virus. In 2009, it infected 10 to 20% of the global population and claimed 250,000 lives. This virus originated from 4 different strains. It evolved through 3 reassortments in birds, pigs, and humans. Moving beyond flu, Let's discuss HIV. Its cross-species transmission is even more astonishing. The AIDS virus is also called HIV, known in Chinese as the human immunodeficiency virus. Once inside humans, it launches massive attacks on the immune system. Initially, the immune system fights back fiercely, eliminating most viruses, but a small number of viruses survive quietly, hiding within specific immune cells. Most of the time, they lie dormant. Once activated, they launch surprise attacks, releasing swarms of viruses to infect cells. The immune system repeatedly counters these assaults. Over time, these surprise attacks exhaust and eventually overwhelm the immune defenses. This process might take just 1 year or up to 20 years, but that day inevitably arrives. Once immunity fails, even common colds or fevers can become life-threatening. HIV wasn't discovered until the 1980s. It took about 30 years to trace its origins. They originated from apes. Scientists, based on genetic backgrounds, HIV is classified into type 1 and type 2. Type 1 primarily originated from chimpanzees in Cameroon. Type 2 mainly came from sooty mangabes in West Africa. Both viruses repeatedly jumped between humans and apes during evolution. From apes to humans, then back to apes. Type 1 jumped 4 times. Type 2 jumped 9 times, meaning today's HIV evolved through 13 cross-species jumps. No one knows exactly how these 13 jumps occurred. In Africa, many keep sooty mangabes as pets and hunt or eat chimpanzees and monkeys. Exposure to their blood transmits the virus. Originally, the virus existed only in Africa, but by the early 20th century, as human migration activities increased, the medical name for this disease is severe acute respiratory syndrome, also called SARS. The SARS virus is a type of coronavirus. Initially thought to originate from civet cats, later research showed civets were only intermediate hosts. The true source host was bats. A SARS virus gene bank was found in bats in Yunnan, proving the virus recombined through multiple strains in bats. This suggests bats in Yunnan infected civets, which were then sold to Guangdong. Transmitting and mutating among civets, it eventually produced the highly contagious SARS virus that infected humans. Civets aren't the only intermediaries. Middle Eastern camels also played this role. A decade after China's SARS outbreak, Saudi Arabia saw another coronavirus in 2012, MERS, Middle East Respiratory Syndrome. The MERS virus came from African bats. These bats first infected camels in North Africa, then transmitted to humans through camel nasal secretions. Similarly, Malaysia's Nipah virus spread when pig farms were built near bat habitats. Bats dropped partially eaten fruit into pig pens. Pigs ate it, got infected, then passed it to humans. Whether consuming wild animals or encroaching on their habitats, humans create conditions for viral transmission. To prevent future pandemics, scientists are studying animals, especially bats, to track viral footprints. But the sheer scale of this work is daunting. In Indian flying foxes alone, scientists identified 55 viruses. 50 were previously unknown. We don't know which might cause the next plague. This leads us to question humanity's track record against viruses. Can we completely eradicate viruses? In truth, modern medicine has only fully eradicated one human virus from nature, smallpox. Over 3 millennia, smallpox likely killed more people than any other disease on Earth. From 1400 to 1800, Europe lost 500 million people to smallpox per century. Only in 1977, after Ethiopia recorded the last case, did smallpox vanish from nature. Humans were able to eradicate smallpox because it only infects humans, not other animals. Systematically eliminating it from humans did the trick. But this doesn't mean humanity is free from smallpox threats, as 2 labs, one in the US and one in Russia, store live smallpox virus, and no one can guarantee it won't leak. The World Health Organization faces a dilemma, whether to order immediate destruction of the lab stocks. Proponents argue it should be destroyed right away. If the virus leaks or is used as a bioweapon by terrorists, the consequences would be disastrous, since people no longer get smallpox vaccines. Immunity to smallpox is fragile, but opponents say that with the risk of resurgence, we need more research to develop better vaccines and drugs. which requires live smallpox virus. No clear consensus has been reached on this, but tech advances have shifted the debate. Gene sequencing tech allows us to artificially synthesize viral genomes. We've already created poliovirus from scratch. This means that while we can't eradicate many viruses, we've mastered the ability to create them. In this sense, these viruses will never vanish from Earth. That wraps up part 2, where we covered how viruses cross species barriers and bring disasters to humans. But that's not the whole story. Viruses also have a lovable side. Now, let's dive into part 3 to explore why humans can't live without viruses. Viruses' first role is shaping our genes. We usually think genes are the most fundamental identity markers of humans. However, the human genome actually contains tons of viral gene traces, numbering in the thousands. Scientists have discovered a type of virus called endogenous retroviruses. Endogenous, as the name suggests, means it originates from within the organism. Retroviruses' RNA can synthesize DNA and integrate into the host cell's DNA, altering its genetic blueprint. And the host will be infected for life with this virus and pass it on to offspring. Once a retrovirus is carried by the host and inherited through generations, it becomes an endogenous retrovirus. Endogenous retroviruses may have infected the common ancestor of mammals as far back as 100 million years ago. They've been repeatedly invading our genomes over time, accumulating to astonishing numbers today. Each of our genomes carries nearly 100,000 fragments of endogenous retroviral DNA, accounting for about 8% of all human DNA. Additionally, there are millions of small DNA fragments that may also be related to the evolution of endogenous retroviruses. The author says that our genomes are practically flooded with viruses. For a long time, people thought they were junk in the genome, possibly even traitors that might sabotage us someday.
But in reality, these viruses have been quietly playing unique roles.
Without them, we might not even be born. For example, one endogenous retrovirus synthesizes a protein called syncytin, which only appears in the placenta and helps the fetus absorb nutrients from the mother.
Scientists experimented with mice by deleting the syncytin gene, and as a result, no mouse embryos survived to birth.
Scientists have found syncytin in all placental mammals. Thus, it's inferred that about 100 million years ago, the ancestor of mammals was infected by an endogenous retrovirus, acquiring the earliest syncytin protein and evolving the first placenta, all thanks to viruses. The second role of viruses is enriching nature's gene pool. Scientists have discovered 1.8 million viral genes in the ocean, and surprisingly, 90% of them are completely unknown to us. The reason for such rich genetic diversity is that viruses act like couriers, delivering genes everywhere. As mentioned earlier, retroviruses can deliver genes to hosts. Similarly, viruses can copy host genes onto themselves and carry them elsewhere to pass on. For viruses, it's like grab and go. Thus, each year, marine viruses transfer about 100 quintillion genes between different hosts. That's 10 to the 24th power. All living organisms today have traces of viruses embedded in their genomes. Genes shuttle between species, profoundly influencing all life on Earth. To give the most direct example, the oxygen we breathe is related to viral gene transfer. There's a bacterium in the ocean called Prochlorococcus, responsible for most of Earth's photosynthesis. Its ability comes from a virus that gave it a protein gene capable of capturing photons. Rough estimates show about 10% of Earth's photosynthesis occurs through viral genes. That means 1 in every 10 breaths you take contains oxygen gifted by viruses. Viruses acting as gene carriers have shaped life's vibrant diversity today. As the author puts it, organisms are essentially just DNA constantly mixing and rearranging. This makes genetic history resemble a bustling trade network, with viruses as the traders. Their third direct role for humans: eliminating bacteria. A virus called bacteriophage specializes in infecting bacteria. It has a sci-fi appearance resembling a lunar lander with spider-like legs beneath its head. After landing on bacteria, it drills a hole, injects its DNA, replicates massively, and drains the bacterium dry. Actually, bacteriophages are closer than you think. They're in yogurt, sauerkraut, and salami, killing bacteria daily. Ocean phages launch 100 trillion attacks per second, eliminating 15 to 40% of marine bacteria daily. Even more remarkably, bacteriophages shape geological structures. Bacteria they kill in oceans transform into rock. For example, England's White Cliffs of Dover formed from massive deposits of dead bacterial shells, creating pure white cliffs tens of meters high. These served as natural navigation landmarks for British pilots during World War II. This landscape was accidentally sculpted by bacteriophages in nature. For humans, bacteriophages' greatest value is disease treatment. Remarkably, as viruses, they only infect bacteria and are harmless to humans. In 1931, bacteriophage medications were developed by the cosmetics company L'Oreal, but using live viruses for treatment made people nervous. It was at this moment that antibiotics were discovered. Even after boiling virus-containing liquid, the viruses remain unharmed. Soak filter paper in virus-laden liquid, let it dry completely, then 3 months later, soak it in water. That water can still infect tobacco plants. To biologists back then, viruses were a completely new life form. Tiny, structurally simple, non-cellular organisms that must parasitize cells. Viruses are totally different from bacteria, another microbe. First off, viruses are way smaller. Regular microscopes can see bacteria but not viruses. Only higher power electron microscopes can reveal what viruses look like. Second, viruses have a simple structure. Bacteria are single-cell organisms, while viruses don't even have cells at all. It's basically just a protein shell holding a few genes. Because viruses are so structurally basic, scientists still argue over whether they can even be called life. Third, viruses parasitize cells. Since bacteria are cellular organisms, viruses can also infect them. Viruses inject genes and proteins into host cells, turning them into factories for replication. A single virus can replicate thousands of copies inside one cell in just a day. Our most direct take on viruses is that they make people sick. Different viruses operate in wildly different ways.